Any questions about anything before we start? Where's Andrew Knaus? You don't know? Oh, I just said, where are you? Because Matt was here, and I thought, oh, where are all the rest of those guys? And then those three came in, and then excuses. Oh. Any questions about anything? First of all, in response to numerous questions that I received after class last time, no, I'm not going to test you on this bio stuff. It's not going to be on the test. And why am I going over this? Okay. Yeah, I know, it's kind of odd. The first day of school, everyone's saying, is that going to be on the test? And, like, oh, don't worry about it. The reason I'm going over this bio stuff is sort of two reasons. First of all, to show you how crazy complex biology and biological evolution are. Second of all, to show you where the inspiration for things like genetic algorithms came from in the first place, and at the same time show you how stripped down the actual genetic algorithms people use are versus how complex the true biology is. And that's supposed to be both good news and bad news. The bad news is that genetic algorithms are so stripped down, how could you ever expect them to solve problems the way biological evolution has? On the other hand, now you see all this wonderful complexity in biological evolution and say, hey, what if with future computing capability I could soup up the genetic algorithms to embody some more of that stuff, to incorporate more of that complexity and maybe do the kind of work that biological evolution does. So that's the only reason we're spending a couple of days on this biology stuff. And I'm not an expert on biology, so those of you who are, I'm probably going to say some wrong things or some slightly, angled off the truth things. But for our purposes, I think it'll be okay. All right, so anyway, where were we last time? And once again, please let me know if I need to stop sooner than the bottom of the board when I'm writing, for those of you who are behind something. So where were we at last time? We were talking about cells in eukaryotic organisms, and specifically somatic cells as opposed to germ cells like a skin cell or a nerve cell or a plant stem cell or a plant leaf cell, whatever. And those things are little chemical factories. Essentially, what cells do is hang out and make proteins. And proteins are these molecules that are the workhorse molecules of all life forms. And last time we talked about the process, how a cell makes a protein, and this business about RNA, transfer RNA, messenger RNA, DNA in the nucleus of a cell. Eukaryotic organisms are cells that have cells with nuclei. And we got this sort of big picture by the end of class last time, that you have this nuclear DNA, and this thing contains the quote-unquote recipes for all the proteins that the cell might ever want to manufacture. And then there's this process where somehow or other a recipe for a protein gets read, and what comes out of that is messenger RNA, a messenger RNA strand with a protein recipe on it. And that comes out of the nucleus and goes out into the sort of the ambient part of the cell, and the ribosome picks that up and reads it like a little cursor, and little globs of transfer RNA come on, and the protein gets formed. So you have your ribosome plus transfer RNA equals protein after reading this thing. So this is a very stripped down picture of how proteins get manufactured. And the process here, where the recipe for a protein gets read onto a strand of messenger RNA, this is called transcription. Okay, that's a word that we didn't use last time. And the process by which the messenger RNA strand gets read by the little clicker in the ribosome and a protein gets produced, that's called translation. So there's two different processes there, and that's what molecular biologists call them. And you can see in this picture, you've got this sort of this flow of information from left to right, from the nuclear DNA to the protein. And like I said last time, this is a lot like a really sophisticated kind of D to A conversion. These recipes are discrete things, written in the alphabet of A, C, G's, and T's. Proteins are like continuous things. They're globules. Their tertiary structures are like the meanings of the proteins, whereas the linear structure of the protein as a polypeptide chain is like a sentence, whose words are these codons, these triplets of RNA nucleotides. And the central dogma, as they call it, of molecular biology says that in a cell, the information flows one way, from nuclear DNA to protein. So the info flow is unidirectional. It's one way, from the nuclear DNA to the protein via this path. And like all dogmas, it's not really quite true. Okay, there's a lot of little chinks in that you have to worry about. And that's why they call it the central dogma rather than the central principle or the founding constitution or whatever of molecular biology. An example of that is you've probably heard of retroviruses, right? What is a virus? Is a virus a cell? Is it a living organism? Anybody know the answer to that? Rishi? It's intermediate, he says, between living and not living. I'd say it's more on the side of not living. But anyway, virus is really just a bunch of RNA or DNA or whatever, just nucleotides of various kinds in a string. And it's kind of like a terrorist or a pirate. You know, it goes into a cell and it hijacks the chemical factory. It's like, you know, look at me, I'm the captain now. That kind of thing, right? Okay, I think that guy's going to get the Best Supporting Actor award. But anyway, aside from that, okay, that's what a virus does. It goes into a cell, and what it does is it sort of pushes aside all this and says, okay, ribosome, make copies of me. Here's the recipe. And so the cell gets busy making copies of this virus. And then the new copies go out and invade other cells and do the same thing. That's how viruses invade a body. But a retrovirus actually affects the nuclear DNA of the cell. It doesn't sort of do it in the ambient air. And retro, the part of that name of retrovirus, is in some sense saying that the virus causes information to flow retro to the central dogma. Okay, so that's a very sketchy description of one of the holes in the central dogma. But anyway, okay, that's what's going on. Now just some numbers, put some numbers to this. How big are proteins on the average? Viewed as amino acid chains. So if you unwrap the tertiary structure and you just look at the linear structure of the protein as a chain of amino acids, how long is that? And it turns out there's quite a range. It's like anywhere from about 100 to about 10,000 amino acids. And an example of a protein, and I always forget whether it's 146 or 141, so I wrote it down someplace. It's 146. A famous protein, hemoglobin, is 146 amino acids long. And of course, hemoglobin is very important. I mean, it's the thing that enables the red blood cells to carry oxygen out to the rest of your body. So it does a very important function, and it's on the short side for proteins. So size is not necessarily the most important thing about a protein in terms of the importance of the job it does. Okay, another thing. Another number kind of thing. How much nuclear DNA is there? Like, remember, you can sort of think of the nuclear DNA as just a big, long double helix, and we'll get more into this in a second, into the formatting, so to speak, of the nuclear DNA. But how many base pairs does a typical organism's nuclear DNA have? Okay, so how big is the nuclear DNA in terms of number of base pairs? And let me just write a number of nucleotide pairs. I didn't really use the word base last time. Well, you know, the numbers are pretty big, but the way that people often talk about this is if you think about each, say, A, T, G, or C as a word, then how many Encyclopedia Britannicas, you know, is the nuclear DNA and so on. I don't want to get into the absolute numbers, but just to give you an example, an amoeba, so the number for an amoeba, of course, is a one-celled thing. It's one of the simplest, I'll put it in quotes, eukaryotic organisms, is on the order of 2,500 times the number in a human. The number of base pairs in an amoeba's nuclear DNA is that much more than a human. Now, is an amoeba more complex than a human? Maybe it is in a sense, because the amoeba has to do all this stuff with just one cell, right? Whereas we have like lots of cells to work with, right? I don't know. But so once again, we don't have a simple mapping between size of the nuclear DNA and complexity of the organism. Okay, so that's just another number thing. And one way to think of the nuclear DNA in ECE terms is sort of like a read-only memory. And I'll just put up ROM, assuming everybody knows what ROM stands for. It's like sort of the brain trust of the cell, right? As long as you believe the central dogma that nothing can go in there and mess up the brain trust, it's like a read-only memory. It can't be written on. Retroviruses can go in and mess with that read-only memory. So in a sense, not only are they pirates, terrorists, they're hijackers. They're hackers, I mean. Okay, so you can think of the nuclear DNA as a ROM. Where does the nuclear DNA come from? Remember, we started with proteins, and we saw how where they came from. We found that in the nuclear DNA of the somatic cell, you have a recipe for all the proteins. Where does the DNA come from? Well, now we have to talk about that a little bit. And before we do that, I want to introduce this very sort of controversial term that doesn't really have an exact meaning. Okay, so first of all, terminology. We call the nuclear DNA, which is the same in every somatic cell. I said that last time, but I just want to remind you of a given organism. The genome of that organism. So sequencing an organism's genome is figuring out what all the A, T, Gs, and Cs in the nuclear DNA of that organism are. Genome has a sort of this GEN part in it, and so the question arises. So I'll put this phrase in quotes: the genome equals the set of all genes, and I'll put genes in embedded quotes, of the organism. And the question arises, what is a gene? And you've all heard the word gene before. And I claim that there's really no satisfactory answer, but we pretend that there is when we talk about things like evolution, when we talk about things like genetic algorithms and all that. So we pretend on a regular basis that there is. And you'll see more about that in a moment. If I say there's no satisfactory answers, I probably ought to volunteer a few plausible seeming answers and tell you why they're not satisfactory, right? So here's some unsatisfactory answers. To that question. First one is a gene is a contiguous strand of nuclear DNA that encodes a protein. Now that's a reasonable kind of definition, right? In some sense, the struggle to define genes is the struggle to atomize the nuclear DNA. Okay? So, if the main purpose of the nuclear DNA is to tell the cell how to make proteins, why not volunteer this as a definition of a gene? That it's a contiguous strand of nuclear DNA that encodes a protein. Well, the problems with that, there's at least two problems. First of all, it turns out that the recipe for a protein need not appear in a contiguous strand in the genome. It might be in several places, and it turns out that the DNA transcript or an RNA transcriptase or whatever it is that peels the DNA apart to make the messenger RNA, to make the protein, might have to go to different parts of the genome to figure out, get the different pieces for a given protein. So it turns out that one protein's encoding might be sort of spread out or distributed over the genome. And I apologize for the bad writing on the board. I'm out of practice the first couple weeks of school. I'll get back into it. Please don't worry. Can you read it? Is it all right? Okay. Turns out that one protein's encoding might be distributed over the genome and might not be part of a contiguous strand. Another problem is that there's a lot of the genome, in fact 99% of the genome as it happens, doesn't encode for proteins. And you might say, well then what does it do? Were you here last time?
Yeah.
Oh. Yeah. Well, you were in South America over break, right?
Yeah.
Lucky you. Wow. I've never been to South America. I'd like to go. Yeah, Machu Picchu and all that kind of stuff. What? We could go on a field trip, yeah. All right. Yeah, well, I mean, that's where Darwin found all this stuff, you know, Galapagos, all that. Hey, we could justify that, couldn't we? Could we? Yeah, Albanese's really nice, you know, he's a really laid back. He might fund it. Okay, anyway, so 99% of the genome doesn't encode for proteins. Well, what does it do? What does it do if it doesn't encode for proteins? People used to say there's only 1% of the genome that encodes for protein, and the other 99% is junk DNA. That's what people used to say. But they don't say that anymore, because what used to be called junk DNA turns out to be rather important to the structure and function of organisms. So it turns out that this stuff does other important tasks. For example, it might tell some piece of this 99% of the genome that does not encode for a protein might tell another part of the genome whether to produce a protein or not. And in some sense, it acts as a switch. So acting as switches that sort of turn on or off whether, and I'm being really loose with the language here, a protein gets produced from a recipe in the 1%. Okay, so this is another version of the 1% versus the 99% that happens in biology. And then there's things that switch on and off the switches. So acting as switches of the switches. Telling the switches whether to turn on, turn off. So there's all these meta levels and complexity that no one really understands completely. It's a big mystery. People are looking deeply into this to figure out what the given control system in a given cell looks like. Now, as it happens, these functions turn out to get performed differently in different kinds of cells in the same organism. And that's what makes, for example, a hair cell different from a nerve cell, different from a muscle cell, is that different genes get turned on, turned off, different proteins get produced, and the way that biologists talk about this is that in different cells, different genes get expressed. And once again, they're using this word gene for which we have no good definition. So different kinds of cells, different genes get expressed in different kinds of cells. Okay, so that's one. Those are two problems with that definition of a gene. That functions of things on the genome are not limited to recipes for proteins. There's all these control system type things going on as well. So what's another problematic definition of what a gene is? So number 2, a gene is a piece of the genome that corresponds, or that quote unquote in writ large encodes, I'll write it in caps because this is not a simple kind of encoding like a recipe for protein, a trait of the organism. And when we talk about the stripped down models of evolution that people talk about in books, popular books and everything, we'll see this kind of thing coming up quite a bit. Okay, now what's the obvious question? Well, what's an obvious question that arises when I write that problematic definition down? Yeah?
Is there a one-to-one correspondence between the gene and the trait?
Well, Alex asks if there's a one-to-one correspondence between the gene and the trait. Clearly no, and we'll talk about that later on. But even more basically, Randy, what is a trait? What is a trait? And in particular, what is a relevant trait? You know? So this begs the question, what even is a trait? Okay, so I'm as clueless about that as anybody is, right? And also, even if you had a definition of a trait, the traits that are relevant to evolution are going to be the ones that are relevant to whether the organism gets to reproduce and all that sort of thing. So anyway, those are examples of problematic definitions of what a gene is, and we don't. The bottom line is, I'm not going to try to define what a gene is. So there's no solid definition for the word gene. But we still think of, and we picture the genome as, quote unquote, an array of genes. of some form or another. And I'm using the word array to kind of feed into the next little bit, because if you think of the DNA, if you think of the nuclear DNA as a read-only memory, one natural question to ask is, how is that read-only memory formatted? Okay, so here's a question. How is the nuclear DNA, how is the ROM formatted? How is the ROM formatted? You can think of it as one big long linear array of genes if you want, but it turns out that in most organisms, well, all the organisms that we know of, the nuclear DNA has a given format. And it turns out that there are two main kinds of organisms that you encounter in real life. Okay, so here's two kinds of eukaryotic organisms. One of them is called haploid organisms. And what is a haploid organism? A haploid organism is an organism whose ROM, whose nuclear DNA, is formatted into a bunch of linear chromosomes that are distinct from each other. So the nuclear DNA is formatted, that is to say, divides up into a finite number of quote-unquote sub-ROMs called chromosomes. Each chromosome is just a piece of the nuclear DNA, a strand of DNA, a long DNA molecule. And there's no sort of a priori relationship between the formatting of these individual sub-ROMs. Like, one could be really short, one could be really long, and so on. So there's no necessary relationship between the formats, quote unquote. And when I say formats, I mean essentially lengths of the different chromosomes. And remember last time I said every time you see that subword somatic, whatever, that means body. And so you may ask, well, where did the word chromosome come from? Because chromo, we all think of that as being like color, right? So colored bodies, what does that have to do with chromosomes? Well, it came from the fact that they were able, when they first discovered the way that the nuclear DNA was formatted, and this is even before they knew about DNA or the structure of DNA or anything, the way they did it was by putting dyes into cells that made the chromosomes turn colors so that you could see them under a microscope. So that's why they call them chromosomes. All right, diploid organisms, same idea. The nuclear DNA is formatted, divides up into a finite number of sub-ROMs called chromosomes, but now the chromosomes come in what we call homologous pairs. Okay, so the DNA divides up similarly. But instead of having no a priori relationship between the formats of the individual chromosomes, the chromosomes come in essentially, and I'm putting this word in parentheses, and I'll ask you if you know why in a minute, what we call homologous, that is to say, identically formatted pairs. Okay, so anybody know whether humans are haploid or diploid? Diploid, yes. And how many essentially homologous pairs of chromosomes does a human somatic cell genome contain? 23 pairs, correct. So there's 46 chromosomes total, and they come in 23 essentially homologous pairs. Now why do I say essentially? So humans have 46 chromosomes in 23 pairs. Why do I say essentially homologous pairs? Rishi? Yeah, okay. So if you're a female, the pairs are homologous. Okay, so all pairs are homologous pairs. They're not homologous with each other, but the two chromosomes in any given pair are homologous. In a female, but one pair, the X and Y, is not so in a male. And there's all kinds of cool stories about why, X and Y, where'd they come from, what's up with that. What is the Y chromosome? It's really just a shriveled up, dysfunctional old X chromosome. Essentially, there was an article in the New York Times like 2 weeks ago, a new story about the Y chromosome, a new story about the X chromosome. And it turns out that, that people's bodies, like a female body, for example, has 2 X chromosomes in every cell, but different cells in the body use a different one of the two of the pair. So it's not, I don't know, it's really crazy. All right, so anyway, that's how the nuclear DNA is formatted into chromosomes. Now, where does the nuclear DNA of a given organism come from? It depends on how that given organism reproduces. And reproduction can either be asexual, which means that every organism has one parent, or sexual, which means that every organism has 2 parents. And I'm going to consider only that case because that's the one that comes up when you do genetic algorithms and stuff. So whence comes an organism's nuclear DNA, ROM, whatever, from its parents or parents. And so put parent with an apostrophe, with a parentheses S. And when you have an asexually reproducing organisms, one parent gives rise to one offspring. And sexually reproducing organisms, 2 parents give rise to one offspring. And of course, I mean here that one parent could give rise to many offspring, but it can do it all by itself. 2 parents can give rise to many offspring, but each offspring has 2 parents. Okay, that's the point of sexually, and we're going to focus on sexually. So we're going to focus on this one. And we're going to look at what happens. First, we're going to look at what happens in haploid organisms, then we're going to look at what happens in diploid. And again, this is all going to be very stripped down. I will be unapologetic about that. But I'll keep reminding you of it. All right, so let's look at the haploid case first. You have 2 individuals, and they're going to make an offspring. So you're given 2 individuals. Say their nuclear DNAs, because I'm going to show you one for one and one for the other, look like this. And I'm going to try to avoid using letters that we've already used. So this is very schematically, I'm drawing this. And I'm drawing on this let's pretend kind of thing. We think of the gene as, or we think of the genome as an array of genes. suppose that one parent looks like B, D, F, H, J, Z, T, S, Q, like that. And it's divided up into chromosomes. So say B and D are one chromosome, F, H, J are another chromosome, Z, T, S, Q are another chromosome. And the other one looks like, and I'll write down in a second what this symbol means, B prime, D prime, or let's use bars because they're easier to see from a distance. F bar, H bar, J bar, Z bar, T bar, S bar, Q bar. OK, so the idea here is you think of the letters, think of each letter as a gene, and think of each chromosome, or think of each bracketed sequence of letters as a chromosome. OK, so the point is that a given haploid organisms that can reproduce with each other, that are of the same, what they call species, they're going to have homologous, they're going to have the same chromosomal structure in some sense. They're going to have one chromosome that has like 2 genes on it, you know, that kind of thing. And once again, this is, like I said, it's a very stripped down picture. It's an array, thinking of the nuclear DNA as an array of genes, each gene represented by a letter. And B and B bar, people would say, terminology, B and B bar are different, what they call alleles of the same gene. And they may be the same. OK, but anyway, I don't want to get too deeply into the bio stuff. So what happens when these guys get together and produce an offspring? Well, one thing that happens is that they produce the nuclear DNA of their offspring by what is called crossover. OK, so the nuclear DNA of the offspring has the same chromosomal structure, the same format, but the individual letters that appear in the different chromosomes could come from either parent. So the parents' DNAs cross over, and I'll show you an example of that in a second, to produce the actual offspring's DNA. So here are some possible nuclear DNAs for the offspring. So here are some possibilities for the offspring's DNA. OK, one possibility would be this, BD, and then F bar, H bar, J bar, and then ZTSQ. So in other words, the offspring has exactly the same first chromosome and last chromosome as the top parent, and it has the middle chromosome of the bottom parent. And you can think of this as being achieved by what we call two-point crossover. You look at these 2 points along the genome, and you cross over, you swap what's between the 2 points between the 2 parents. Okay, so this is achievable by two-point crossover. And so you may ask, when crossover occurs, does it respect the boundaries between chromosomes? No, Max is right. It does not. OK, crossover does not respect the boundaries between chromosomes. You could also get BD, and then say F bar H bar J with no bar, and then ZT S bar Q bar, like that, something like that. So note crossover disrespects boundaries between, well, that's a bad way to put it. It doesn't take whole chromosomes and swap them for whole chromosomes. So it can occur anywhere in the genome, not just at interchromosomal boundaries. Okay, here's a final possibility. Say, how about BD FH bar J bar Z bar T bar S bar Q bar. This one has the first 3 genes in the genome, which constitute the genes in the first chromosome and the first gene in the second chromosome, from the top parent, and the rest from the bottom parent. And as you can see, you can think of that as being achieved by 1-point crossover. You go to this spot between the F and the H, and you take this much from the first parent and the rest from the second parent. And as it happens, that's the main kind of crossover that people use in genetic algorithms, is 1-point crossover. OK, so this is achievable by 1-point crossover. So you may say, or ask, you may say, OK, well, hey, so this is how the parents make the genome of the offspring. How do they decide where to cross their genomes over? Who decides that? Whose chance? Again, I forget. Yeah, there's no rule that says, hey, we're going to cross over here from now on, right? That the way that the crossover occurs is random. So this is an important point. Crossover occurs randomly. It might not happen at all. In fact, you might have an offspring whose genome is identical to that of the parents. So you might even have, and I'll break my promise, I'll put one more offspring DNA. You might just have the barred one, B bar, D bar, F bar, H bar, J bar, Z bar, T bar, S bar, Q bar as the offspring's DNA. So there's no crossover at all. And this is one of the important sources of randomness in evolution. And it's a way that genetic diversity gets introduced into a population. And now you realize, here, I'm talking about a genome that's only 9 genes long with 3 tiny chromosomes. That's nothing like a real genome. You can imagine the richness of the source for diversity in life that happens when you have bigger ones. OK, now another thing can happen en route from the parents to the offspring. Another random effect. And actually, there are other random effects, but I'll talk about one other random effect because that's one that comes up in genetic algorithms. So one other important random effect that can occur en route from parents. And I'm going to be very explicit here, from the parent's nuclear DNA to the offspring's. I'm not talking about from the parents to the offspring because we're Max. Yep.
Like documented cases of there being no crossover in like human.
Question is, are there any documented cases of no crossover in humans? I don't know. I mean, I've seen people who look amazingly like their mom or their dad or whatever, but But the thing is that even if you have an identical genome with somebody, it doesn't mean you're the same person. So I don't know. Yeah. Well, I don't know. I don't know. OK. And that is this idea of point mutations. OK. So for example, a B bar, you know, what might happen when these things cross over is that the B bar gets copied incorrectly into a B prime or something. So for example, B bar becomes B prime, which is not equal to B or B bar. Or at worst, it becomes something completely non-functional. So, or even worse, B bar becomes something non-functional. Okay, so how do point mutations happen? Well, the source of these point mutations, there's lots of sources of it. So one source of mutations is the kind of the chemical similarity between the different DNA base pairs, the ones we call the purines and the ones we call the pyrimidines. Okay, the purines, which remember are the ones that don't have a Y in their name, so they're adenine and guanine, are very similar. They're, in some sense, they're two stable states of almost the same. And once again, please, if you're a bio expert, don't take me to task because I'm oversimplifying things. Chemical structure. And similarly, for the pyrimides, T and C. They're both stable states of an almost same molecular structure. And you've all seen courses, you've all been in courses where you have stable states and you have a potential energy barrier, something like that, right? Is that familiar? Kinda? 30-30? No? Quantum? Did you ever hear about that, Vinay? No? Maybe you're the wrong one to ask. No, just kidding. All right, well, you have in the world, you have things that are like low energy states. And we'll have a lot more to talk about low energy states later when we talk about the Metropolis algorithm. But you have low energy states separated by potential energy barrier. And all it takes to knock something from one of those into another is enough energy to vault you over the potential energy barrier. And what does it take to vault you over the where does that energy come from? Ionizing radiation turns out to be something that can do that. So radiation can vault you, so can vault an A into a G and vice versa. And that's why, for example, that's why radiation is dangerous in some sense. Okay, that's why, you know, for example, a friend of mine actually, he had to have radiation treatments, right, for something, and he's still young, he's still of childbearing age, but they didn't want him and his wife to try to have children for at least a year after his last radiation treatment, because what could have happened during his radiation treatments was that some of his A's could have been vaulted into G's, some of his T's could have been in his germ cells. We haven't even talked about germ cells yet. And they wanted to have that get all out of his system before he tried to reproduce again. Okay, so that's an example. Another kind of randomness that comes into this reproduction process. Okay, and that's kind of those things are like the inspiration for most of what happens in genetic algorithms. But let's talk about diploid for a second because we're diploid, and Actually, now's a good time to take the 3-minute break. I have to get used to when to do the 3-minute break at a class that starts at 1:25. So let's do it now, and then we'll talk about diploid for a second. All right. So anyway, this is a very stripped down picture. Very stripped down picture of how the nuclear DNA of two parents produces the nuclear DNA of an offspring for a haploid organism. Now I want to look at a very stripped down similar picture for diploid. Okay, so let's look at the same thing for diploid. Okay, so the idea here is that how do two parents' nuclear DNAs, and this is terrible sentence grammar, DNAs, that's a horrible thing to put on a board, but you know, what the heck. Nuclear DNAs give rise to the nuclear DNA, the genome of an offspring. Okay, well, diploid organisms have some special purpose organs that produce, and remember we're focusing on sexually reproducing organisms where you have two parents per offspring. You have special purpose organs produce what are called germ cells. Okay, so what are germ cells? Germ cells, as opposed to somatic cells, are cells that are exclusively for reproduction. So, for example, in humans, eggs and sperm. What does a germ cell look like? A germ cell looks kind of like a haploid cell, a haploid somatic cell. So a germ cell, quote unquote, looks like a haploid somatic cell in the following sense, at least genome-wise. Okay, what a germ cell in an organism has, it has one chromosome for each pair of chromosomes in the organism's somatic cell. So it contains one chromosome per pair of chromosomes. Let's put it this way, somatic cell chromosomes. So to put some numbers to this, in a human, a human germ cell contains 23 chromosomes. Okay, so every sperm in a male contains 23 chromosomes. Every egg in a female contains 23 chromosomes, and each of these chromosomes is homologous to the chromosomes in one of the pairs in the somatic cell. Okay? Where do these come from? How do these chromosomes get produced? So, how do these get produced? Well, they get produced from the genome of the organism that produces them. And in the special purpose organs that produce them, what happens is this process called meiosis. So a process called M-E-I-O-S-I-S, meiosis, makes them. And how does that happen? Suppose that in your somatic cell genome you have chromosome pair A, for example. So no, let's not use A, let's use 1. Suppose in the somatic genome, you have, say, pair number 1 of chromosomes. Okay? What you want to do is you want to produce, for a germ cell, the chromosome corresponding to pair number 1 in the somatic genome. So you build a quote-unquote chromo number 1 for the germ cell by doing this kind of haploid type crossover between the pair of homologous chromosomes in pair number 1. And I'll draw a little picture in a sec. If my awkward attempt at describing this in English is bad. So what's the last word over there? Build a chromo number 1 for germ cell by crossing over haploid style the pair of chromosomes in pair number 1 and producing one offspring chromosome, and then plants that in a germ cell as chromosome number 1 in the germ cell. Pair number 2 does the same thing to make chromosome number 2 in the germ cell, and so on. So for example, if the somatic genome has 2 pairs of chromos, okay, what happens is, so you have pair number 1 is, let's say, gamma 1 and gamma 1 bar, and pair number 2 is delta 1 and delta 1 bar. Okay, so this is chroma number 1 pair, chroma number 2 pair. These two cross over to make the gamma for the germ cell, and these two are the gamma prime for the germ cell, and these two cross over to make the delta prime for the germ cell. So this is the germ cell genome, and this is the somatic genome. Okay? Now the cool thing is that, as you know, this crossover can take place in any number of ways, right? So all your different germ cells are going to have all different kinds of gamma primes and all different kinds of delta primes. Okay, so all your germ cells have all different kinds of, say, gamma primes and delta primes, depending on how this meiotic crossover occurs. And don't forget mutations. They can happen too. Point mutations. So there's yet another layer of random process that happens here when you have a diploid organism. You have this crossover taking place in the production of germ cells. And what happens when the parents mate, however they do that, is that one germ cell from one parent and another germ cell from the other parent come together. And does anyone know what, in the process of reproduction, what do you call a germ cell that's in action? There's a name. I think this is right. A gamete? Is that right? And then they fuse to form a Zygote, yeah. Okay, I have it right. Okay, good. So when the parents decide, or whatever, when the parents produce an offspring, one germ cell from each come together. That sounds awkward, but you know what I mean. And they make one diploid cell, which is the first cell of the offspring organism, whose nuclear genome, remember the nuclear genome, the chromosomes comes in pairs, has a pair, each of its pairs is going to be one member of the pair is straight from the father, one member of the pair is straight from the mother. Okay, so whose nuclear genomes pairs each consist of one chromosome from each parent. So for example, if this is what the two germ cells look like, one of them has gamma prime and delta prime, and the other one has gamma double prime and delta double prime, then the offspring's genome looks like this. Gamma prime, gamma double prime as one pair, and delta prime, delta double prime as the other pair. No further crossing over or whatever happens there. So in some sense, like if you look inside your cell, right, you being a diploid organism, you'll find that your somatic DNA, each chromosome pair, one of those comes from your mom, one of those comes from your dad, biological mother, biological father. That's what I'm talking about here. Okay, so straight up, modulo whatever mutation has occurred. Okay, so that's the way the offspring's genome arises from the parent's genome. Now, remember, this is just how does the nuclear DNA, how does the genome give rise to the genome? You've answered that question, and we can see that this process contains a lot of sources of randomness. Crossover, what kind of crossover? Where do you do it? Is there a mutation? There's others. There's things called inversion, and there's whatever, deletions and all that kind of thing, all kinds of other stuff. But you can see now that in any case, the parental genomes turning into the offspring genome, this process has lots of random effects. From crossover and mutation and everything. And as it happens, this is the best understood piece of randomness in all of biology. That there's so much other random stuff that we don't understand at all, but this kind of randomness is the most well understood. And when I talk about evolution next time, we'll see what I mean by that. Okay. Now, one of the sort of leitmotifs here, the sort of the sub-stories, is that every child of two parents starts off as a single cell. And the only info is the nuclear DNA in that single cell. And somehow that becomes this whole creature, right? Okay, so note, the offspring starts as one cell, and its nuclear DNA, its genome, let's use the word. Its genome contains all the info, and I'll put this in quotes, necessary to build the offspring as an organism. Okay, now there's a word that people use that I hate when they use it. Okay, and Melanie Mitchell actually uses it on page 5 of her book. Okay, and I'll read you that passage in a minute. But the word I hate that people use is blueprint. Okay, they say the genome is like the blueprint for an organism. So here's a fallacy, a fallacious sort of commonplace as a noun. Quote unquote, the genome is the blueprint for an organism. Okay, why is that wrong? What is a blueprint? A blueprint, they hardly even use those anymore, I bet. Like architects are all like computerized now, but they used to have these big tables where, you know, they want to build Duffield Hall, right? They make these diagrams very carefully by hand, and they have these big like scale models of whatever's going to happen in the building. And the ink is always blue, and so they call it a blueprint. So a blueprint is like a scale model of something. The genome is not a scale model of an organism, right? What is it? It contains all the info necessary to build an organism, but in that sense, it's not a blueprint, it's a recipe. If anything. And even that is an oversimplification. Even that gives the genome too much credit. So here's better than this, but still too simple. Quote unquote, the genome is the recipe for an organism. Okay, so what's a recipe? A recipe, you know, like say you're baking a cake, right? This many cups of flour, this many cups of sugar, this many quarter pounds of butter, you know, whatever. Or margarine, don't use this, don't use that, whatever. The recipe could be the same, but if you use like, you know, weevily flour or sour milk when you make the cake, it's going to come out different, right? Okay? So a recipe does not determine the organism. If you have good recipes, a good recipe plus, say, bad ingredients, or perhaps an inaccurate oven temperature thingy. It says it's 450 when it's really 275. Or high altitude, low atmo pressure, whatever, equals maybe a bad cake on one day, but the same recipe plus good stuff gives you a maybe, depending on whether your nephew is making it or your mom, okay, a good cake. Okay. Now, what's the point here? The point here is that there's this great book, and on the website I put a whole bunch of books that you might want to like look at one day. If people, does anyone read books anymore? Or is it all TLDR? Yeah? Okay. There's this really cool little book. It's very easy to read. It's by this guy who's a little bit doctrinaire, but anyway, in his own way, Richard Lewontin. It's called Biology as Ideology, the Doctrine of DNA, where he goes about deconstructing this whole notion of, you know, everything's genetic and blah, 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 and really, one of the points of emphasis is this whole recipe versus blueprint thing. Identical twins have the same genome. They have the same recipe for an organism. Everyone here probably knows a pair of identical twins, right? Maybe, or has known them. Are they the same person? No. Are they identical in every way? No. And that's because different stuff happened, right? All right, so we'll get more to this next time. Yeah, what? What's that?
I thought only fraternal twins were not identical.
They're not genetically identical.
Oh, really? I thought like everything was the same.
Do you know any